souvent, nos collègues étrangers sont un peu surpris parce que c'est la parole. Donc, traditionnellement, on est sur une vingtaine de minutes par personne. Donc, je jouais un peu le modérateur pour le temps. Et je lui donnerai la parole un par un. On lance le passage à la discussion sur les techniques. On commence. Donc, Sophie et Oscar, les animateurs, les modèles. Et ensuite, les super sympathiques. Alors, sachant que nous, en général, ce sera plutôt voilà. Je vais passer avant vous. Donc, on retrouve Oscar et Sophie. Je sais pas. Oui, voilà, c'est ça. Ça va. Ça devrait être donc, de passage, Sophie, Oscar, Gérard, ouais. I'm so sorry, I had 2 weeks. No worries. We had pretty well. Hello, Sophie, I'm Fred Young. I will be the president for this jury. So we have a little talk before you join us to organize the discussion after the defense. So I don't know if you are familiar with the defense in France, but usually it's quite short. We have something like 20 minutes to discuss with the candidates. And because most of the jury is through video conference, we are not going to make a kind of general discussion among all the members, but we will do it the more classical way. Each member is in turn. And so, if you don't mind, ladies first, as Oscar said, we will start the discussion with you. It's okay with you. Okay. Yes, I've got lots of questions. Yes, it's really frustrating usually because I will have to probably cut you after 20 minutes to listen. It's really as compared to other countries. Yeah. So I will before it's very briefly us. So just and then we will have this discussion meeting. So first Sophie and then go there, myself, Stephanie, and then. And then after we meet together, and go. And Sophie, so you were associated professor at the University of Edinburgh. Okay. And the research was in population genetics and user methods do you want to add something else? No, I think that's and it's Emilie Day. Yes, I did.
Tot wat dient die webcam?
Ja, normaal gezien kunnen daar de jury mee zien.
Ah, oké.
En dat is de webcam die mij ziet.
Zware angle nu te zien. Zo.
Echt, maar ik kan deze uitzetten.
Of aan beneden zo. Oké. Dus de mensen via YouTube, die zien dat.
Ja, normaal gezien.
Oké, is goed. Maar dat is zo'n rare angle.
Ja. Maar ik denk dat ik dit zelfs wil uitzetten.
And we are going to start in 12 minutes.
Hi, so I hope you can all hear me correctly. Awesome.
Yes, perfect.
de votre présence. C'est agréable de voir aussi la soutenance de thèse de retour, même si aujourd'hui, cette soutenance de thèse, donc Emilie, à Boulanger, nous allons écouter dans une seconde, aura lieu en partie en présentiel et en visioconférence. Plusieurs membres du jury sont en visioconférence, vous pouvez voir l'écran. Cette soutenance de thèse, elle aura lieu également en anglais. Je fais juste l'introduction en français et ensuite nous passerons en anglais pour la présentation et les discussions, parce que tous les membres du jury ne sont pas francophones. Alors, le jury m'a chargé d'être la présidente, j'ai essayé de faire ça. Donc, je me présente, je m'appelle Frédéric Emilie, je suis directrice de recherche au CNRS. Je travaille à l'Institut des sciences de l'évolution de Montpellier, sur le campus de la PAC. Et mon domaine de recherche concerne l'utilisation des outils ADN pour étudier la diversité et la dispersion des organismes marins. Je continue en vous présentant les membres du jury. Nous aurons donc deux référés, deux rapporteurs, qui sont d'une part Sophie van der Velen, elle est là, Sophie, je pense, qui est professeure associée à l'université de Stellenbosch et dont la spécialité est, j'ai décidé de également des approches environnementales. Le second rapporteur est Oscar Kodla, professeur au Leibniz Center, dont le domaine de recherche concerne la génétique et la génomique de la population d'organismes marins. Ensuite, Robert Bunier, directeur de recherche à l'Institut d'océanographie Paul Ricard, et il interviendra sur son domaine d'analyse environnementale et de génétique de population d'invertébrés et de carnivores de façon générale. Et enfin, les deux encadrants de cette thèse, d'une part Stéphanie Manel, directeur d'études à l'école pratique et méthode d'études, travaillant en serre, et dont le domaine de spécialité est la génomique des populations d'organismes marins. Et enfin, Daniel Nouveau, professeur à l'Université de Montpellier, au laboratoire Marseille, et dont le domaine de recherche et d'expertise est en écologie marine. Voilà, je vous remercie beaucoup. Je vais donc laisser la parole à Emilie pour sa soutenance. Je pense que je vais 45 minutes.
C'est ça.
Et puis ensuite, on entamera la discussion avec les membres du jury. Emilie.
Okay, so thank you. And thank you all for being here today as I present and defend my thesis about how we can inform marine biodiversity conservation using next generation sequencing tools, going from genes to communities. So biodiversity is the cornerstone of life, and changes to biodiversity is inherent to this life on Earth. However, changes have been accelerating in this recent history, and we are globally seeing a loss of species worldwide as more and more species are going extinct, as well as a loss of populations and with it within species diversity. Now, marine ecosystems host an abundance of life with over 200,000 species already described. And many of these species represent important sources of proteins and income for humans around the world. However, this incredible diversity is also faced with increasing anthropogenic threats, such as overexploitation, as an estimated 34% of marine fisheries are currently overexploited, as well as the increasing threat of climate change. And as temperature is one of the leading factors determining species distributions worldwide, as temperatures and heat waves increase due to climate change, species that want to avoid local or global extinctions will either have to adapt to their new conditions or move to more favorable conditions. Now, the Mediterranean Sea in particular is a global hotspot of biodiversity. Despite representing less than 1% of the world's ocean's surface and volume, the Mediterranean Sea hosts between 4 to 18% of all marine organisms in the world, and many of those are endemics. However, this incredible biodiversity is also at an arms race with the increasing anthropogenic pressures. As the Mediterranean Sea is a hotspot of pressure, it's one of the most overfished seas in the world, with 74% of the fish stocks overexploited, and also one of the seas knowing the most important threats of climate change. And for example, the Mediterranean Sea is a semi-enclosed sea with important gradients of temperature from north to south and west to east. And now, because of these semi-enclosed natures, many species, and mostly cold adapted species, might see themselves trapped in these colder regions as temperatures increase if they cannot adapt. Now, to face these increasing anthropogenic impacts, marine protected areas are more and more established. So there are delimited areas at sea under a certain management plan, and they can have different levels of protection. And now the strongest form of protection is offered by no-take MPAs, or no-take reserves, where every form of fishing, many visitation practices are prohibited, all of this to limit direct human impacts on marine ecosystems. Now, the establishment of MPAs has been increasing globally. We are currently at around 7% coverage. All of this to reach the goal of protecting 30% of the ocean surface by 2030. Now, in the Mediterranean Sea, a good amount of marine protected areas have already been established. However, as you can see here, most of these protected areas are not or even poorly protected. And when we look at the truly no-take marine reserves, these represent only 0.06 of the whole Mediterranean surface. And most of these no-take MPAs are located in this northwestern part of the Mediterranean Sea. So a lot of work is still needed to reach this goal of 30% coverage. And we need to be able to better understand biodiversity patterns, to know where to place these MPAs, and better understand the effects MPAs have on marine biodiversity to keep improving our conservation measures. So MPAs have been studied for decades, and many of their benefits are already well known. For example, MPAs and reserves in particular are known to increase the density and biomass of fisheries targeted species. However, much less attention has been given to the species richness component of biodiversity. Several studies looking at it find that species richness increase inside of reserves, but this effect is much less pronounced than the effect on density and species abundance. Others find no overall effect on species richness, and some even find that species richness is lower inside of the no-take MPAs than outside. And one of the reasons for this could be that not all species will react the same to protection. So where fisheries targeted species might find refuge inside of no-take reserves, other species that are not targeted by fisheries might find that the areas under disturbance outside might release new niches or might release them from a certain predator pressure. And so to be able to understand the effect MPAs have on this global aspect of, or the total aspect of biodiversity, we need to be able to understand what the effect of reserves is on the species richness and composition of communities when we take these whole communities into account instead of focusing on species targeted by fisheries. Now, at the same time, marine protected areas and reserves can also protect the mechanisms that sustain biodiversity, such as dispersal and gene flow, when they're implemented in connected networks. So many marine organisms have a dispersive larval phase, during which they can be transported by oceanographic currents. And by protecting, for example, source populations or key connectivity nodes, we can protect this dispersal and ensure the dispersal between MPAs, and then ensure the recruitment in other populations against recruitment fluctuations or against overfishing and depletion due to overfishing. At the same time, protecting dispersal allows us to protect the gene flow between populations and thus the maintenance of genetic diversity. And considering this gene flow between populations allows to also preserve the evolutionary adaptive potential of populations against climate change. So for example, if we have two local populations that are differently adapted to their local temperature regimes, protecting this locally adapted population with warm adapted genotypes would allow them to further spread these warm adapted genotypes through the network and preserve the population and its persistence in the long run against climate change. However, understanding where to place these MPAs means we need to understand how Mediterranean populations are connected or inversely structured and where there is prevalence of local adaptation, especially in the Mediterranean Sea. So to answer these two questions, we need adequate tools to measure biodiversity. Now, on the one hand, the richness and composition of diversity and communities is often measured with visual methods, for example, underwater visual senses, where divers will often target fisheries targeted species, and they will either explicitly or implicitly miss a large aspect of this marine biodiversity. For example, they might miss these cryptobenthic fishes, which are gobies and blennies that are typically hiding under crevices and under rocks, or pelagic species that are passing overhead or right before. Now, to understand population structure and connectivity, well, it's difficult to track a small larvae directly. So genetic methods can allow us to study the realized connectivity and the population structure. However, marine organisms inhabit this large, vast open ocean with few physical boundaries and have high capacities for dispersal, leading to high levels of gene flow and thus potentially little genetic differentiation. And when using only a few genetic markers, it can be difficult to discern this genetic structure, as well as to find patterns or look for patterns of local adaptation. So my thesis is centered around answering these two main questions. So what is the effect of protection on species richness when looking at these whole communities, and how are Mediterranean populations structured? And for this, we can benefit for answering these questions, we can benefit from the advent of next generation sequencing. So next generation sequencing allows the parallel sequencing of millions of DNA strands at the same time and offers us new tools to study marine biodiversity at its different levels. At the level of the species richness and species communities, we can leverage environmental DNA metabarcoding to identify all species present in a given environment. And at the level of populations and genes, we can genotype single nucleotide polymorphisms all across the genomes of individuals and thus have thousands of genetic markers to look at genetic structure. So environmental DNA refers to this DNA that is excreted, so intra or extracellular DNA excreted by organisms into their proximate environment. And by extracting and sequencing this DNA, we can identify all species present. So the identification of species is done by sequencing a specific barcode region, which typically are flanked by two highly conserved primer regions or invariable primer regions, which are identical between different species of our target group. So in our case, this will be the teleost fishes. And then in the middle, we have this highly variable barcode region, which varies between different species of our target group, and thus allows us to identify these different species. So in my thesis in particular, we use the 12S metabarcode, 12 teleo metabarcode. And so a general environmental DNA metabarcoding study consists first of collecting environmental samples, extracting this DNA, and then amplifying and sequencing this barcode, which then generates millions of sequences generated through high throughput sequencing. These are then cleaned up and clustered together and compared to a reference database of all known sequences, or ideally all known sequences of our targeted species, which allows us to identify these different species and finally study the richness and composition and other ecological factors of our communities. And in my thesis, I will mainly focus, or I participated and conducted the fieldwork and environmental DNA collection part of this workflow. I focused on building or completing this reference database for species identification and then conducted ecological analyses. So at the level of populations, next generation sequencing allows us to sequence these single nucleotide polymorphisms, which are variants at single sites and occur very frequently across the genomes of organisms. And by sequencing these SNPs, we can obtain thousands of genome-wide genetic markers to have the necessary resolution to look at the population structure of marine organisms. And because they are distributed all across the genomes, these SNPs could also be positioned on protein encoding regions and thus on genes potentially under local adaptation, under selection, and thus indicative of adaptive processes. So typically, SNPs are obtained by collecting tissue samples directly from our organisms, which are then fragmented using restriction enzymes. And these millions of fragments are then sequenced thanks to NGS methods. Now, these millions of fragments can then be aligned against a reference genome to look for these variants at single sites. And by analyzing these allele frequencies at our single sites, we can further look for population structure and, for example, find barriers to dispersal and different populations, as well as look at how these vary with environmental variables under a seascape genomics approach, and look for patterns of local adaptation to environmental variables. And in my thesis, I mainly conducted the final analysis of this workflow, although I was also involved in some of the SNP filtering. So my thesis is structured around three main chapters, which each have been published separately in scientific journals. So in this first chapter, I leverage environmental DNA metabarcoding to look at the community richness and composition level of biodiversity. In a second chapter, I use SNP genotyping to look at the population connectivity across the entire Mediterranean basin. And then in a final chapter, I again use SNP genotyping data to look at the structure and then the local adaptation patterns of different species across the Mediterranean Sea. And so during this presentation, due to time constraints, I will mainly focus on this first and third chapter of my thesis. So in the first chapter, I look at the effect protection has on the diversity and structure of fish communities. So as I already explained in the introduction, we are globally seeing an overall decline of biodiversity. However, this global decline of species does not mean that species richness also necessarily is decreasing at a local scale. And for example, several studies have looked at time series data of species richness in local cases, and they found that in many of these local cases, species richness is actually increasing over time. And this can be explained by a combination of invasions and range expansions, and actually illustrates an interesting paradox, namely that diversity and species richness can remain unchanged or even increase under human-mediated disturbances. Now, marine protected areas offer a unique opportunity to study this paradox from a spatial context, as well as some of the underlying hypotheses. And here, the application of environmental DNA then allows us to identify all these species, going from the smallest, often not targeted by fisheries, to the larger species and those targeted by fisheries. And this environmental DNA metabarcoding can then help us answer the question as to how species richness varies between protection levels. So to study this, we went out and collected environmental DNA from 6 marine protected areas and no-take reserves across the or along the Mediterranean, French Mediterranean coastline. And sampling consisted of collecting four replicates of 30 liters of seawater, each time inside of the marine reserve at 5 kilometers outside and 10 kilometers outside. Now, this DNA is then extracted from our filters, sequenced, cleaned up, and compared to a reference database to identify our species. And these reference sequences are typically obtained from public databases such as GenBank. However, at the onset of our study, the 12S Tilio barcode that we were targeting was only present or only referenced for 31% of all Mediterranean fish species. And these represented only 37% of the local species pool. So a big part of my PhD was to start the completion of this reference database, thanks to the help of a lot of students in the field and in the lab. And then by 2020, we managed to sequence a total or add sufficient sequences to this database to reach a total of 41% of Mediterranean species, but these represent 75% of the local species pool. And although this work is still ongoing, these 75% of species do equally represent the different trophic levels and size classes among our species and are thus not expected to bias our results. So over the 18 sites and 72 samples in total, we identified a total of 97 species and 43 families. And I then calculated the species richness as well as the community composition and dissimilarity in community composition between our different sites. And I compared these different metrics across protection levels, so inside marine reserves at 5 kilometers outside and 10 kilometers outside. But now in the Mediterranean Sea and marine ecosystems in general, habitat and environmental variables play an important role in structuring communities, and this could not be overlooked. So I also collected various habitat variables, such as the percentage coverage of seagrass, rocks, sandy bottom, as well as chlorophyll as an indicator of primary productivity. We calculated the bottom depth, distance to coast, and temperature. Now, this yielded a lot of variables, and because there were too many and to avoid problems of collinearity and overfitting, I then conducted a principal component analysis on these habitat variables and retained only the first 4 PCA axes, which explains 74% of these habitat variance. Now, looking at how species richness varies between our protection levels while taking these differences in habitat configuration into account, we found that marine protection and no-take reserves indeed have an effect on species richness, and this effect was indeed paradoxical, because we found that species richness was actually lowest inside of the no-take reserve and increased with distance outside of the reserve, and was even highest at 10 kilometers outside. Now, this change in species richness could in large part be explained by a change in the diversity or richness of cryptobenthic species, so these gobies and blennies we saw earlier, as their richness almost twice and almost 3 times increased at 5 and 10 kilometers. However, this increase in richness was not simply an increase in similar communities, but more or less species. But we found that we also had a turnover of these different communities, and the different protection levels hosted different compositions of species. So to study and formally test this, we conducted a multivariate model, where, as you can see here, every point is represents a different sample and a different site, and the different species that was found at each of these sites. And here we see that sites from the same level of protection tended to be closely together in their composition and the species we could find than sites outside at 5 or 10 kilometers. And so this result tells us that indeed protection has a significant effect not only on richness, but also the composition of communities. And we found that the sites outside, at the left side of this first axis, mostly represented larger species of higher trophic level and more vulnerable to fishing, whereas the sites outside of the no-take reserves mostly hosted smaller species of lower trophic level and generally lower vulnerability to fishing. And interestingly, we also found mostly pelagic species frequented no-take reserves, whereas outside we again mostly found those cryptobenthic species. So overall, these results tell us that indeed reserves do not harbor more species, but they do harbor different species than the fished areas outside. And one of our hypotheses, or our hypothesis to explain this difference in communities, is that fishing induces a change or could induce a change in trophic interactions. Because these larger and higher trophic level species are typically the ones mostly targeted by fisheries. And because they tend to disappear outside of no-take reserve, their increase in density inside could increase a predation pressure on the lower trophic level species. And these lower trophic level species, that are typically smaller and often those cryptobenthic species, then find refuge outside where the predation pressure is inversely reduced. Now, this signal could then also be uncovered thanks to the detectability of environmental DNA, which allowed us to identify an average of 30 species per sample, which is a lot if you're used to diving in the Mediterranean Sea, and also allowed us to detect these cryptobenthic species and pelagic species that are often missed by visual methods. Now, in a second part of my thesis, I looked at the population structure and adaptive potential of species in the face of climate change. So different species have their environmental niches to which they are best adapted. And the environment can also play an important role in structuring within species population structure and diversity, as different populations can be differently adapted to their local conditions. Now, as marine organisms typically have these high levels of gene flow, this could limit their potential for local adaptation. However, more and more studies are increasingly finding marine organisms such as corals, lobsters, and fishes to be locally adapted to environmental conditions such as temperature, salinity, or productivity. And understanding these patterns of local adaptation, especially in the Mediterranean Sea, is important to assess the vulnerability of our species and populations to climate change and adapt management strategies accordingly. So we set out to understand the population structure and local adaptation patterns of two economically important fish species in the Mediterranean Sea, the white sea bream, Diplodocus cervus, and the striped red mullet, Mulus homiletus. So both species are highly targeted by small-scale fisheries in the Mediterranean, and they have similar distribution ranges across the entire Mediterranean basin, as well as the Northeast Atlantic. Now, both species have similar levels of similar pelagic larval durations, so we can expect similar levels of potential larval dispersal. But the sea bream is capable of more and more widespread adult migration and movement than the striped red mullet, which is more sedentary. And because of these different levels of adult dispersal, we were interested to see if they also presented different levels of genetic differentiation and whether this potential differentiation was mostly driven by neutral processes such as gene flow or processes pertaining to selection such as local adaptation. So to disentangle these different processes, we needed to sample individuals across wide spatial scales and wide environmental gradients. And so for this, I could rely on the extensive individual-based sampling that was conducted as part of the C-Connect project, which was part of Alicia Dallongeville's PhD before me. And here, a large fieldwork team collected individual samples from sea bream and mullet across almost the entire range of the Mediterranean basin. And thanks to next-generation sequencing and SNP genotyping, we could then genotype thousands of genetic markers for 297 individuals of sea bream and 467 individuals of striped red mullet. And now, because we expect our species to have these high levels of gene flow, we wanted to augment even more this resolution of our genetic markers. And so I further selected the most highly differentiated SNPs, which allow us to augment the signal that is generated by neutral processes, such as the signals of population differentiation, as well as capture SNPs that are potentially under selection and thus illuminate adaptive processes as well. And this yielded a total of 413 highly differentiated or outlier SNPs for our sea bream and 291 highly differentiated or outlier SNPs for the striped red mullet. Now, in the first step, I assessed the genetic structure between individuals, and then I formally tested whether this genetic structure could be explained by a combination of geographic distance and larval connection probabilities that we obtained thanks to the model of previously published by Andrello and collaborators, which would then, these signals would be signals of neutral processes, or indicating neutral processes, such as gene flow, or whether our structure could be explained by environmental variables, such as salinity, productivity, or temperature, which would be indicative of local adaptation processes. For the white sea bream, then, we found that individuals could be divided in a total of 3 genetic clusters or populations. So here, every line represents an individual, and every color represents one of these genetic populations they are assigned to. And as you can see, most of the individuals of the Mediterranean Sea form one large genetic cluster, except for these individuals here in the Alboran Sea and along the North African coast. And now this area actually coincides with the area where the fresher and colder water from the Atlantic flows into the Mediterranean and meets this warmer and saltier Mediterranean water, forming a nearly permanent oceanographic barrier to dispersal and a biogeographic barrier that was previously detected for many other species to be a biogeographical barrier between different populations, but was not yet detected for our sea bream. And so formally testing confirmed that indeed this pattern could be explained by isolation because of larval dispersal and geographic distance. But at the same time, these genetic differences between our 2 main groups could be explained by differences in salinity and temperature, indicating that this differentiation between our populations could also be maintained by divergent adaptation to these different salinity and temperature regimes between the Atlantic and then the Alboran and the Mediterranean on the other hand. For our sea bream, we found more pronounced population structure, and we found between 3 to 7 genetically distinct clusters. So the structure is most visible when we look at these 3 clusters. And here we see that we again have this separation of the Alboran Sea individuals, but we further have a gradual separation of the Western Mediterranean and then the Eastern Mediterranean. And again, we find this effect of the Almeria-Oran front, this barrier to dispersal, which formal testing confirmed that our signal, and formal testing confirmed that our longitudinal signal could be explained here by a combination of, again, larval isolation and geographic distance, but also by differences in summer temperature, which could indicate that our signal is a result of a combination of maybe neutral processes such as isolation by distance, but also gradual adaptation to these warming temperatures as we move eastwards. Now, interestingly, we also found that several individuals from 3 groups here in the Alboran Sea, the Northern Gulf of Lion, and the Adriatic Sea genetically actually grouped together and were more similar to each other than these individuals in between. And testing showed us that this signal was mostly explained by changes in primary productivity as well as colder winter temperatures. And when we look again at the maps of our Mediterranean Sea, we see that indeed, these 3 areas coincide with levels of high productivity and cold water temperature. And because this signal breaks with the gradual signal we could expect under isolation by distance, we interpret this that it is very likely that these populations are locally adapted to these colder water temperatures. And interestingly, a similar signal was recently also detected in another Mediterranean species, the ocellated wrasse, where sampling along the Spanish coast found that individuals from the northern Spanish coast and thus the Gulf of Lyon, and then the southern Spanish coast and the Albertan Sea, indeed were genetically more similar to each other than individuals in between. And the authors attribute this also to different levels of productivity and water temperature. So then to further investigate this local adaptation nature of our species, we took these highly differentiated or outlier SNPs and looked at how they were positioned on the reference genomes of the published genomes of our species. And we then selected those markers that were located on protein encoding genes. And we found for the sea bream 5 markers, and for the striped red mullet 7 different markers that were positioned in protein encoding genes and were significantly correlated to temperature levels. And now several of these markers were located in genes that were previously identified in other marine species to be involved in the temperature and heat shock response of these species, thus again indicating that we have processes of local adaptation at play. So overall, as a result, we can say that the gene flow and local adaptation processes interplay differently to shape our species population structure. So for the white sea bream, we found only little genetic structure within the main Mediterranean basin, which could be explained by the species' higher potential for adult dispersal and thus higher levels of gene flow, while the striped red mullet had a stronger differentiation within the Mediterranean Sea and even signals of local adaptation, which could be explained by its more restricted adult dispersal and more restricted gene flow. And again, these signals could be uncovered thanks to the high resolution obtained from single nucleotide protomorphisms. So overall, these next generation sequencing tools allowed us to uncover new patterns of biodiversity of marine fishes. At the levels of communities and compositions, we found that reserves hosted less but different species and mostly higher trophic level and larger species, whereas outside we mostly found smaller species. And then at the level of the population structure and adaptation, we found little structure for the white sea bream within the main Mediterranean basin. However, we found more pronounced differentiation and local adaptation patterns for our striped red mullet. Now, these new patterns of biodiversity also imply have new implications for conservation. So the lower species richness we found in marine reserves did raise some eyebrows when we first published and communicated these results. However, these results do not mean that MPAs do not work, because even though they host less species, they host significantly different species than their proximate outside. And in doing so, they contribute to increase the overall regional diversity of marine communities. Now, these results were recently corroborated by a study of Loiseau and collaborators, who looked at the diversity of birds, tropical reef fish, and alpine plants between 3 levels of protection. So they looked at protected areas, partially protected, and unprotected areas. And they found that diversity patterns and communities indeed significantly differed between these different protection levels, and each level of protection hosted their unique set of species. And so together, these results tell us that a combination of fishing pressure and marine reserves then lead to different levels of pressure and thus heterogeneous fishing pressure, and which we could also obtain by combining different levels of protection, such as partially protected areas. And together, these heterogeneous pressures then create heterogeneous communities, thus increasing the overall diversity of marine communities. At the level of our populations, our results could then be integrated in MPA management to give an evolutionary perspective to this MPA planning. So we found in the northwestern Mediterranean, these populations that are likely adapted to colder water temperatures. And these populations can then be more vulnerable to the increasing temperatures under climate change. However, these areas also coincide with areas of the Mediterranean Sea under the highest fishing pressure. And because we cannot locally halt climate change, putting these populations under the protection of reserves would then allow them the time to adapt potentially to these higher temperatures and give them a fighting chance against the double threat of climate change and fishing pressure. On the other side of the Mediterranean, we found that populations might be then adapted to higher temperatures, and protecting these areas MPAs and no-take reserves would allow us to conserve these warm, potentially warm-adapted genotypes, which could then spread throughout the Mediterranean and increase the overall evolutionary and potential and the persistence of our populations against climate change. Now, environmental DNA has shown to be this incredible tool to gain a new perspective on the effect of protection on species richness and composition. But several improvements can still be made to make this a suitable tool for MPA, or a better tool for MPA monitoring. So on one hand, much is still unknown about the spatial-temporal dynamics of environmental DNA. For example, experimental studies in aquariums find that DNA can be detectable in the water for several hours up until several days. And modeling studies then show that this DNA can be transported over several to 40 kilometers. However, empirical studies find increasingly heterogeneous signals of eDNA and are able to differentiate sites and habitats only a few meters to hundreds of meters apart. So there's still a lot of discordance between these experimental results and then the empirical results. I think more attention could be given to this to better understand the spatio-temporal resolution of our signal and then the spatio-temporal resolution of our results. Now, MPA, one of the most sought after results of MPA protection is often also this increase in density and biomass of our fisheries targeted species. However, our environmental DNA metabarcoding data, as it was implemented in this thesis, could not give us information about the abundance of our species. And we only looked at the presence and absence, because different species will have different excretion rates, which might also differ between different life stages. Different environmental conditions can then affect the persistence of DNA in the environment, and the stochasticity of the amplification process adds even more variability to these results. However, making it difficult to extrapolate species abundances from metabarcoding data. However, more and more studies looking at species-specific DNA signals find increasingly a strong correlation between their species abundance and their eDNA signal. And thus, future work or a promising approach to obtain abundance data from metabarcoding data could then be to calibrate species-specific and sample-specific eDNA concentrations to be able to understand the abundance of species from metabarcoding data. Now, I think what would be interesting would also be to gain a wider understanding of the effect MPA have on our species communities. So here we looked at MPA effects in temperate reef systems, but we could also, for example, replicate our study in other systems such as tropical MPAs to see if the same rules about species assemblages still apply. Now, at the level of our populations, the implications for conservation I formulated rely on this signal of local adaptation we found. And although we found some candidate genes to local adaptation, which give us more confidence in this interpretation, we still did not formally test whether these populations were indeed better adapted to their different environmental conditions. And so a next step would be to conduct experiments, for example, by taking individuals from this potentially cold adapted population in the coldest margin of the Mediterranean Sea, as well as individuals from this potentially warm adapted population, and put them under the same climate conditions and see if either populations indeed have different vulnerabilities to temperature increases. Finally, comparative analyses are incredibly valuable to understand the effects, for example, protection has on different species with different life traits. As well as the effect of environmental variables on different species, for example, with different dispersal abilities. And here, between our two species, we found that even relatively small differences in their adult dispersal could lead to larger differences in population differentiation. And so what would now be interesting is to cast a wider net for species traits and include even more species in our comparative analysis, for example, more sessile species that are not at all capable of adult dispersal, and see how these populations are differentiated across the Mediterranean Sea, and whether we also find more or less patterns of local adaptation. So I have a lot of people to thank, and first of all, the members of the jury for being here today and having this discussion with me later on. But mostly I had these amazing past 4 years with my fair share of ups and downs, I think, but most of all, I learned that I have this amazing support system on which I can count, and that made me look back on these past few years with nothing but a huge smile on my face. And for this, I would first of all like to thank my amazing team of supervisors, Stephanie and David, not only for making this research possible and this thesis possible, but also for supporting me throughout. And I would like to thank you for all the effort you've put into this project. I would like to thank my my team of close collaborators, with whom it was a pleasure to work and from whom I've learned so much. I'd like to thank all the people involved in the fieldwork aspects and the sampling of this thesis, collecting samples from species and from environmental DNA. And I would also like to thank the BEV team in the widest sense of the word for being just amazing people and making it a pleasure to come into work each day. And then finally, I'd like to thank my family, my friends back home, and then all my friends I've made in Montpellier who's made this new city also a home, and my housemates in particular. So thank you, all of you.
Very much for this very nice presentation, summarizing very well the main outcome of the.
Thank you.
We will just wait for people to move or. Okay. Are you? Yes, they are there. Oscar, Sophie, and. Sophie, Oscar, can you hear me?
Yes.
Yes, quite faintly. I could hear Emilie very well, but Fred, it wasn't so easy to hear you. So
we do have a microphone.
it must be here.
And here, Sophie, can you hear me better?
Fantastic. Thank you so much.
Okay, nice. Okay, so, we are going to start the discussion with the jury members. Usually, I like when the discussion is across all members with the PhD candidate. However, because of the video conference, we are going to make it more classical. So each member of the jury will discuss with you in turn. And so we start now with Sophie van der Eden. So please, Sophie, start the discussion with Emilie.
Fantastic. So before I ask you some questions, Emilie, I just wanted to congratulate you on a very tremendous and very fine piece of work that you've produced. It's really a fantastic PhD. So congratulations to you and your supervisors. It's very interesting that you have them as the kind of top predators in the ocean on your thank you slide. So I love it. So yes, Emilie, so I've got quite a few questions, so we'll see how we do for time. Are you able to hear me quite clearly?
Yes, I hear you fine.
Super, okay. So I hear a little bit of myself with the transmission, so I'll speak a bit more slowly and maybe have to wait occasionally, okay. So I guess my first question is a bit more general. So you spoke about species extinctions on your first slide, and you know how a lot of this is driven by anthropogenic change and climate change. But are there any recorded marine species extinctions in the Mediterranean?
Can you please repeat that for me? I didn't.
Okay, I will do, no problem. So on your first slide, Emilie, you spoke about marine species extinctions, right, as a result of anthropogenic drivers and pressures and climate change. But tell me, have there been any recorded marine species extinctions in the Mediterranean in recent history?
So total species extinction, I don't honestly really know. But I know that we are, for example, seeing turnovers in species assemblage in this eastern part of the Mediterranean as water temperatures are increasing, and more species, for example, are arriving from the Red Sea and locally replacing communities.
Okay, sure. All those Lesepsian migrants. That's right. They
Yeah.
make things a bit more difficult. So I kind of wanted to, pick up on that a little bit with regards to your eDNA metabolic coding chapter in particular. Do you think that eDNA is a useful tool to understand extinctions going forward? How would you, and if you had to design a study, or design really a monitoring protocol to understand species extinctions in the Mediterranean, how would you do that? What would your ideal study protocol be?
So I think environmental DNA is definitely an environmental DNA metabarcoding is definitely a very valuable tool to do that, because it has this advantage of creating this very standardized protocol that can be easily replicated over time and by different samplers, and then with little sampling bias. And then how I would implement that is I would, integrate a regular sampling protocol. So species monitoring, maybe targeting certain species. So either we could target entire assemblages and look at how these presence absence of species at a certain location vary over time, or we could maybe target certain species and look at even how their abundance and their eDNA abundance signal varies over time, and see if we see any differences as time progresses.
Sure, that would work. I think you've hit very importantly on the time component, right? The temporal aspect of this, I think, becomes very important. So you're entirely correct there. And just to kind of carry on a little bit with the eDNA metabarcoding, I was really fascinated, I think, as many of us were when your paper was published, that showed these really quite strong community turnovers between the different levels of protection. So do you think that you can potentially use environmental DNA as a tool to monitor the effectiveness of marine protected areas based on what you've shown already?
So I think, first of all, we'll have to redefine what it means to be an effective MPA, because as our results shown that looking at the species richness is not the best indicator to say that species or MPAs protect species are efficient or not. But then I think we could indeed monitor, for example, these presences of our target species that are known to be vulnerable to fishing and reliable on conservation measures to be adequately protected. And so I think environmental DNA metabarcoding would then allow to monitor their presence in marine protected areas and reserves, again, at different sites and different protection levels. I don't know if that really answered, yeah.
Yeah, it does in a way. I just think that if you show these really high levels of community structuring, different protection levels, you might be able to use a similar signal, from protected areas around the Mediterranean to kind of draw some conclusions about, whether they are effectively being protected or not, right? So on one of your slides, you showed where they are protected areas and whether they are no-take or, what really their level of protection and really enforcement is. And I think it's about enforcement. But I think, environmental DNA, from what you've shown, could really be a very useful tool to monitor the enforcement of marine protected areas by kind of, by picking up on these community structures. So yeah. And I'm going to actually
so I want to say something about that, because I think indeed we could then use environmental DNA to maybe calibrate some sort of species communities, and then maybe define the communities typically found inside of the no-take reserves or typically outside. And here we didn't look at these intermediate protection levels, but we could also look at these and then find, for example, this panel of species typically found inside or outside, and maybe check their presence in different MPAs under different enforcement levels. And indeed, we could then maybe monitor whether these enforcement levels are effective and or at least comparative to other reserves under similar levels of enforcement and protection.
Yeah, that sounds great. And I think that's something that I think is definitely one of the more powerful aspects of environmental DNA metabarcoding. So we can talk about that going forward. But yes, so just one other question. Well, I've still got a few, but what I wanted to know, so you showed that in the Northern Adriatic Sea in particular, there's quite a dominance of colder water, and that there's also quite kind of elevated levels of primary productivity. Can you explain to me a little bit about the oceanography of that region, and what the mechanisms are behind those oceanographic patterns?
Yeah, definitely. So the Mediterranean Sea has actually quite a complex oceanography, which is mainly generated by this inflow of Atlantic waters into the Mediterranean Sea, which then creates these circulation patterns going along the African coast and then moving back up. And so in these areas, in the Northern Adriatic and then the Gulf of Lyon, we have this seawater arriving from the warmer regions so this warm surface seawater arriving. And I think we also have very strong surface winds, which creates an upwelling of waters from the deeper areas of the Mediterranean Sea, which bring up these nutrients and makes these regions very nutrient rich and having high productivity.
Okay, so it's very similar to here in South Africa, where it's a wind-driven upwelling system. So, okay, just a lot more localized in the Mediterranean. That's really interesting. Okay, so I've got a few more questions. Particularly, I'm really interested in how we can use genomic tools. For the particularly for looking at where to put marine protected areas and looking at their effectiveness. But that obviously always comes at a balance with the socioeconomic needs of a particular nation. And the Mediterranean is super interesting because there are so many different countries that rely on its resources. So you have very different kind of socio-political agendas that drive the exploitation of the marine resources in the area. So, marine protected areas obviously sometimes can be very effective, and in other regions be not so effective at all. So they're not always necessarily the best tool for protecting things such as commercial fisheries. Can you think of other can you think of other ways that we might protect, certain fish species in particular, without relying on, for example, marine protected areas?
So I think if we want to indeed protect certain fish species in general, then we can, for example, imply fishing restrictions on these species. So a typical example in the Mediterranean Sea is this brown grouper, which was highly targeted by fishing, and especially, I think, underwater spearfishing. And so the numbers in the Mediterranean Sea decreased substantially over time, and then they implied these moratoriums or fishing restrictions. And now they can see that inside the marine reserves in particular, but over time now these populations or groupers are actually reestablishing. And so these fishing restrictions are proving to be efficient in protecting this brown grouper. So I think if we want to indeed target certain species, we can then, imply fishing restrictions on these species while still allowing fishing of other species that are less threatened.
Okay, sure, that sounds great. And then from a kind of taking you back to genomic diversity, right, or genetic diversity as a tool for monitoring, for example, fisheries impacts or kind of, climate change impacts going forward, there are obviously lots of different metrics that you can calculate from a SNP data set. There's a whole bunch of metrics that can be used to look at how diverse the population is. So from a monitoring perspective, so trying to understand how genetic or genomic diversity might change, either with fishing pressure or just in general, with fishes moving in response to climatic change. Have you maybe thought about which metrics in particular, that capture diversity might be the most sensitive and therefore the most useful to monitoring, genomic diversity in space and in time?
So which metric in particular? I don't think I really have an idea about that. I guess, well, for example, when we look at these highly differentiated outlier SNPs, these are the ones that show the strongest effect of our population differentiation. And because they are potentially under adaptation, they are also potentially being influenced the fastest. So maybe looking at these most differentiated regions of the genome allow us to faster detect differences at the level of the population and better track current influences of, for example, population diminishing and losses of diversity by looking at those most differentiated sites, I think. But I don't have really specific metrics in mind.
Okay. No, I was just thinking, I mean, there's obviously lots of ways to do it, and how which metric might be most appropriate will also depend on the species and things such as generation time and life history and those kind of things. But it certainly looks from some of our work that we've been doing that things such as allelic richness, just the number of SNPs, might be a very useful tool. It's obviously always quite difficult in marine systems where everything is moving and it's moving to where you don't know where it's, where it's going to be next. So, but you're right that certainly, there's a lot of promise in putative outlier loci, particularly if we're going to find that, that populations are going to be shifting in responses to some of the environmental changes that are in the Mediterranean. So yeah, that's a solid answer, I think. And then also, I was interested in the two species that you chose. How representative do you think they are of fish communities in the Mediterranean? And if you had to design your PhD again, if you could redesign it, would you maybe add some different species, yes or no, or how might that look?
So I think one of the main advantages of choosing these species was that they were distributed across this entire Mediterranean range. And so this allowed us to look at how this environment played on all our populations. And I think they're quite representative of the various species we can find in the Mediterranean. Now, if I would have to maybe add more species to it, I was thinking that maybe, for example, looking at these species for which we are now currently seeing significant impacts of fishing could be very interesting to more, and because they have more pressing conservation issues. So for example, these groupers that are now almost only found inside marine reserves, I think it would be very interesting to, for example, look at how their populations are also structured, to then allow to see how this fishing pressure could also influence these population structures, and to monitor these species and develop conservation actions that are for species that are more pressing conservation need, I think.
Sure, because with because also with your SNP data, I mean, you could potentially model demographic change through changes in effective population size quite nicely, to really show the impact of things such as fishing. There's some interesting data coming out globally that's been doing that. So, but yeah, grouper obviously under high pressures everywhere globally. And then I guess I don't want to ask too many questions because I know that I need to leave some for the rest of your jury too. So I just kind of want to just finish on one, and maybe I can add something later if there's something else that we haven't touched on as a jury. So I saw in your chapter 2 to detect those putative outliers, you used one program, you used PC Adapt. Now, there's certainly what was the reasoning behind just using PC Adapt? So maybe let's start with that.
So because we actually had individual-based data, and I didn't really show it, but at each site we had between 1 to maximum 10 individuals per site. We didn't have these clearly sampled populations, and so we couldn't use population differentiation methods that are often used. And so we were actually limited to using these methods where we could use individual-based sampling.
Okay, that explains it, because obviously, you know, you can throw 5 different outlier programs at your data and actually get quite, you know, and that every 5 you know, each of those 5 programs will detect, may detect different outliers. So, you know, it's certainly one of the drawbacks, one of the limitations of the approach. It's not, you know, it's not to invalidate it, it just kind of weakens the potential for detecting.
Yeah, I think it would have been great indeed to be able to combine different outlier detection methods to then, you know, look whether they find the same SNPs under selection indeed, and more target these markers that were detected by the different programs and different, yeah.
Sure. And then one last question, Emilie, before I hand over. Can you tell me a little bit about how, you know, about the level of cooperation between countries, you know, with regards to fisheries resources? So here in, you know, to give you an example, in South Africa, we've got the Benguela Large Current Commission, the BCLME, you know, which kind of seeks to kind of open partnerships between Angola, Namibia, and South Africa when it comes to joint fisheries resources. It's a bit more difficult between South Africa and Mozambique because of the level of knowledge and enforcement. You know, so there's a mechanism locally that provides an opportunity to kind of co-manage and come to decisions about the management of marine resources where they are transboundary. Can you tell me a little bit about the setup in the Mediterranean, and if there is, you know, if there is a more collaborative approach, whether it's very much kind of, you know, in kind of just based around individual countries?
So I didn't closely work with the really management side of the Mediterranean fisheries, but what I've learned from looking at different fishing reports is actually I think that the fishing in the Mediterranean Sea is strongly regulated per country and not yet really cooperated between different countries. I might stand corrected here, but I do know that we have this now this METPAN network, for example, which is the network of Mediterranean marine protected areas, which seeks to build a network between different MPA management organizations to be able to better work together and really collaborate on these MPA efforts. However, at the fishing level, I don't really know.
Okay, super. All right. Well, then, thank you for again for a really great talk to you, you know, and for a fantastic PhD to you and your supervisors and your team. And I think I'll hand over to the next member of the jury.
Thank you very much, Sophie. The micro is. Thank you very much. And now, Oscar, we can listen to you, please.
Okay, thanks. So yes, thanks for this very nice presentation. And I really liked, you know, the combination of approach. You didn't present everything that was on your thesis the presentation. I think that's fine to do editorial choices. But so I really liked, broadly speaking, the combination of approaches and questions that you addressed with your thesis, all within a coherent framework. So yeah, my first question, and I don't know, I may be confused about that, but so 4 years ago, I was also on the jury for Alicia's thesis, and she presented her data, and so the big thing was that she had actually pooled data because the coverage on her data was way below actually even 1x. So she had to pool samples per location to genotype them. And so I might be wrong, but so I'm under the impression that we're talking about the same data set that is now magically turned into an individual based data set. And yeah, so I just wanted to just get that clarified.
Yeah, so indeed, so for the striped red mullet, we are talking about the same data set. So the CBREAM data was actually resequenced because it didn't yield satisfactory results. And the CBREAM data, actually, one of the the striped red mullet data, one of the problems was that many of the fragments were partially degraded. And because of the software they used, these fragments could not be used. However, here we, for the SNP calling, we actually use the FreeBIAS software that is not as strict on the length of the fragments required. And that allowed us to, well, mainly Pierre-Edouard Guérin, because he did the bioinformatic analysis. So that actually allowed him to trim the unsatisfactory parts of the sequences and actually keep much more sequences from the species that were initially possible with the first analysis. And then because we had these reference genomes of our species, we could use this FreeBIAS software and then these fragments of different lengths and then keep more of the sequencing data. And I actually looked up the sequencing coverage of our different species. And for example, for the striped red mullet, so this is the data after I've already filtered for a minimum coverage of 3 reads per marker. But we actually have an average of 10 reads per individual across the different markers.
So then, so what you're telling me is that what you're saying is that people are just losing money by sequencing stuff 10x. You could sequence stuff less than 1x, use FreeBase, and then achieve actually a 10x coverage. Is that correct?
No, I don't know about that. I missed the beginning of your sentence, I think.
So if I take that, so considering your answer, so I would say that there is no need to sequence anything at 10x coverage. Why don't we all sequence 1x coverage and take actually this FreeBase approach and end up with 10x coverage of individual samples? So are people losing money by over sequencing their sample? Why don't everybody do what you did?
No, I do think you, so we actually did sequence at sufficient coverage in the beginning, but it's just that unfortunately the sequence yielded many sequences of bad quality. And so, but you did need to sequence sufficiently to have this sufficient coverage, but we could trim these bad quality edges or extremities and then still keep our initial sequencing depth.
Okay. So what you basically did is that you rescued a large proportion of the data by trimming, and that allowed to actually,
And then for comparison, for the CBREAM, we did have much higher coverage.
Oh, that's better. But I mean, I would say that 10x is okay. So I have no issue with that. Okay, cool. Yeah, so I wasn't sure because that was not very clear.
Yeah.
So I wasn't sure whether I missed something but okay, that's clear now. Thanks. And then, talking about the connectivity. So you didn't present too much the connectivity paper. I really liked it.
Thank you.
I was thinking these days, I'm thinking about coalescent connectivity. And so if I think about it, what you looked at is filiation. So parent-offspring relationship and beyond. So basically, if you could have stepping stone between over several generations. But actually one of the results that I found interesting is that at the end of the day, the geographic distance did a better job at predicting the genetic patterns than the stepping stone, more elaborated oceanographic model. But I was wondering if there is maybe something missing when we consider this type of framework. So imagine 2 populations that do not share any migrants. So they don't share migrants. So if you look for you take a direct approach to find migrants, even first or second generation, you'll find none of them. But both of these populations receive migrants from a third population. So they do share alleles. So isn't that also important when we try to infer connectivity? So not just the direct parent-descendant relationships, but also the coalescent aspect. And could that possibly be an improvement?
I don't think I entirely understood. So you mean if they received migrants further back in time and not at the present time anymore?
No, not even. So imagine you have two populations that do not exchange immigrants, but those two populations receive migrants from a third population. So they receive the same migrants, the migrants from the same population. So these two populations, even though there is no direct gene flow between them, there is still, if you want, coalescent gene flow, in the sense that you could go up and find the origin of alleles that you find in the two populations, but that do not come from direct gene flow among them.
I guess this then relates to this problem of unsampled and ghost populations that we did kind of try to incorporate in our project, because indeed, if we don't have this information of this third population, well, we're missing part of this information. And what we tried to do with our sampling model was to equally represent different points along the Mediterranean Sea, to take these unsampled and so potentially third populations into account in this stepping stone process. So indeed, then from a genetic point of view, we can't tell whether our two populations are actually closely related to each other or mostly thanks to this third population. But I think this is what we did try to account for, or what integrating these all potential populations, even the unsampled ones, in our stepping stone model would achieve.
Yeah. So I saw that you took actually, so the model accounts, right, for the possibility of stepping stone through unsampled population. I mean, that's a slightly different thing, but I can see this point. But so I like the idea. So I like the idea of the model that tried to connect, to make the link between just the parent offspring assignment methods to population genetic methods, and that effectively actually links the two. And that's super cool. Then I have a kind of more an open question. So this relatedness. So let's imagine that you have entire genome, so you're not limited in the number of SNPs that you can have. So how far can we push this relatedness approach? So we can probably identify sibling, we can probably identify the half siblings. So could we go further than that? And could we use other signal of the data than just SNPs? So these days in our group, we're thinking about fragments of identity by descent, for example.
Can you repeat that last part? I didn't hear it correctly.
Well, I'm just wondering about the power that we have to infer relatedness from individuals in the wild, especially marine populations that are very large and so have a high effective population size. So how far can we go in terms of relationships among individuals? We can resolve SIBs, half SIBs, but how far can we go, and could the use of fragment of identity by descent help?
So I think here one of the most limiting factors would probably be the sampling part and the sampling sufficient individuals, because we only had about 470, so we found a few individual pairs. But to get a better view of the relationships among all individuals, because we have indeed these large populations, I think we'd have to sample many more individuals. But one of the advantages of then looking at the relatedness between individuals compared to, for example, parentage assignment tests, tests is that it does require less individuals to be sampled than parental analysis, because to assign individuals back to the parent pairs, we need to have almost an exhaustive sample of this parent population. And by looking at the relatedness directly between species, between individuals, we can have more, find more opportunistically find closely related individuals. So I think then to go further into these relationships, maybe if we sample more individuals and could potentially sample these parent populations, we could find more direct assignments.
Yeah, so that's exactly what I'm thinking about. So I'm thinking when you do parent offspring, you need to sequence a large proportion of the parents.
Yeah.
Sure. But then by looking at more distant relationships, it allows you to still find those relationships with a less exhaustive sampling. So what I'm thinking is that to push this reasoning on steroids and say, okay, so I'm not going to worry about sampling exhaustively the population. What I'm going to try to do is to actually do a much better job at identify the relatedness, even the much far away relatedness relationship between individuals.
Okay.
And I mean, could we do that if we have with whole genome data?
Honestly, I don't know. I guess if we look at relatedness as a correlation measure between individuals, we could then lower the threshold. And if we have these full genomes, we have a better resolution at this similarity, at assessing this similarity between individuals. And then we can look at lower similarity coefficients and lower relatedness coefficients to look at relationships further away than siblings or maybe first cousins.
Yeah, exactly. So you can possibly reduce the interval, the confidence interval, to be able to not to exclude zero. Yeah. Yeah, I'm just thinking loud. It's not like these are easy questions. So, so did you find yourself limited, by the way, by the number of SNPs that you had for that, or you thought it was fine?
I don't think, no, we were limited by the number of SNPs. I didn't test with different subsamples of numbers of markers, so.
Okay. And then for the same for local adaptation. So you have between 1 and 8,000 SNPs. So what proportion of the genome is that?
I'd say it's a very small proportion of the genome. Yeah, I don't remember their exact genome size, but I think, yeah, these 8,000 SNPs is a very small proportion of the genome. So.
Yeah, so it's probably like in all these GBS studies, it's probably less than 1% or around 1%. Now the question, I don't have a strong opinion of that. So do you think it's useful, it's sufficient to actually address local adaptation, or do you think we might miss important things, or do you think actually no, if you could just, like you have a few, I think over 100 outliers, that's actually enough to make the point that you're making about local adaptation? Of course, more SNPs maybe increase the chance that you hit a gene that's under selection, sure, but yeah.
So I think it's definitely useful, because we did find this strong signal in the striped red mullet, and we did find these several SNPs that were located on these protein encoding genes and in known genes related to temperature effects. However, we only found 5 and 7 of these, so we were still quite limited. And so among these highly differentiated SNPs, we only found 5 and 7 located in gene coding regions related to temperature. So I think if we increase, the amount of markers we have, we definitely would find, or potentially, if there is more local adaptation and selection, we definitely find more regions of interest.
Yes, but so adaptation to temperature is probably highly polygenic. So you could find more genes, but if they're all part of the same, in the end, metabolic pathway on the same adaptation. So I'm just thinking loud of whether this is actually useful or not, or are you missing? So in our case, in reef fishes, we work on color pattern. We realized when we switch from RAD to whole genome that yes, we missed important loci, but it's because color pattern has a very simple genetic basis with a few loci with a very strong effect. And then these peaks are needle sharp, and it's very easy to miss them, but something like temperature may be different. Yeah. So I have 4 minutes left. I'll just go with your eDNA chapter, which I really liked. And I was really pleased by the results. Yes, it's something we're not used to do. I was thinking about, is it possible that the results that you got are due to the fact that we're dealing with fishes? I mean, fishes are very specific, right? They're highly targeted by fisheries. And do you expect to have the same results if you had done your entire studies on invertebrates? So we have some, I don't know, I can think of the effect maybe of lobsters, of a dense population of lobster that could have cascading effect on the entire ecosystem. But so do you think on invertebrates we could have the same thing or the same patterns, or you think we could find reversed patterns, or it could be totally different? Yeah.
I think it would definitely be interesting to look at these trophic interactions at a wider scale, indeed beyond just the fish species. At the scale of the lobsters, I know that lobsters are highly targeted by fishing as well, because, well, they're very delicious to eat, and targeted by spearfishers as well. And so, indeed, I think I need to more understand the very specific trophic interactions, for example, between lobsters and what their prey are. But it would be interesting to then look at more, how do I say this? So get a wider view of these possible trophic interactions by indeed looking at invertebrates and maybe I'm thinking other species that could be prey of our fishes, for example, and see how their abundances or their presence also varies between protection levels. And their environmental DNA could also be very useful because we now already have these samples and we can actually resequence them many times and actually target other taxonomic groups.
Yeah, agreed. You would just need an army of PhD students to do an invertebrate database. But yeah. So these were my main questions. Let me see if I had anything else. So I think I'll let the next person take over. Thanks.
Thank you very much, Oscar. Perfect timing again. So now Robert Bunet will discuss with Emilie. Can you hear us?
Yes, I can hear you. Yes, thank you. Thank you. So can you hear me well?
Yeah, perfect.
Okay, because there are some road working at the moment on the island, and maybe they will come a bit later close to my window. So if you can't hear me, yes, just tell me.
Yeah, so it's a little bit difficult to hear. So if you can talk slowly,
okay.
yeah.
Okay, so can you hear me better now?
Yeah, better.
Yes. Okay, so I will try to increase a bit my voice. So first, I would like to congratulate you, Emilie, for your very clear presentation and your very well presented manuscript. I have had a lot of pleasure to read your manuscript and listen to your talk. So
thank you.
I would like to also thanks all your colleagues that made this work possible. But today is your day, so it's it's for you. It's your. I've got a lot of questions, but most of them have been already asked previously. So I will focus a little bit more on the methodological things, and I will focus come again on the question of Oscar previously. You focused mainly on fish on your DNA database. And so do you expect you or someone else to maybe look at the eukaryotes, for example, on your DNA that you isolated from your samples with primers targeting the 18F, for example?
To what? Can you repeat, please?
So you limited your studies on fish mainly.
Yeah.
On the eDNA. And do you expect to reanalyze maybe your DNA samples that you have, I think, with primers targeting the 18S?
Oh, okay.
Yes,
So to still target, so different primers, but still to target fish populations, or you mean more look at the eukaryote diversity in general?
Yeah, in general, in your
Yeah.
in your 5 and 10 sites.
So I guess we were not immediately planning on that, but it definitely would be an option, and it would be very interesting again to see how well other elements of the food chain and even other aspects of marine diversity vary, and whether they're also affected or not by this protection level. But we were not immediately planning on doing that.
Okay, so but you still have your DNA samples, right?
Yeah, we still have them, and we can still resequence them.
And do you expect also to, or do you have some other classical monitoring methods to compare with your eDNA analysis?
So we didn't have any other methods that were conducted at the same time, although we do have checklists from the different reserves where they, at other times, when diving and assess. So we don't have data on exactly the same time periods, but we could compare checklists, and we did find different species with our eDNA, so a lot of overlap, but we still find different species between these different methods. And I think this is what we're mainly seeing in the literature as well, is that eDNA captures a wide diversity, but each method still has its specificities, and there's never 100% overlap between the fishes identified, for example, with visual methods or with fishing techniques, and then with environmental DNA metabarcoding.
Yeah. Okay. And I have a little question in your manuscript, because there is something I missed. I think if I go back to your manuscript, you present the supplementary figure S4, page 169, with the overlapping fish in the reserve, 5 kilometers and 10 kilometers outside. So you have 8 species which are typical from reserve, and then I can't really find them in the supplementary figure S5. And if I go to the in the table,
You mean this one?
Yes.
Okay.
But then when I go to the supplementary table S7, you have the Aterina boyeri, which is a pelagic fish and which is unique to reserves.
Yes.
Yeah. And for example, we did some sampling here around on the islands, and we didn't find them very often. We found them only in the harbor of
okay.
So it's not MPA here. How could you explain this? But you don't find them very easily, and when we go to the under the sea, when we dive, we always see this Aterina boyeri fish always. We have a lot of these fish here, but we can't find them in the DNA samples. How can you explain this, maybe?
I guess so. Here it was detected in only one side inside of the reserve, and so this figure in general. So we didn't include it in the main manuscript because it shows the total number of species observed uniquely in each sampling site, and often these were detections in one single site. So not, for example, across all marine reserves. And now the reason why these might be missed inside MPAs is. I guess it might pertain to the time scale of our eDNA signal. And if this is a pelagic species, it's probably not very sedentary and might move a lot around. And if you pass diving at the wrong time, you might miss them. But as DNA stays detectable in the sea for at least maybe a few hours, the eDNA signal might still pick up. It could also be that these species might avoid diver presence. I don't know much about this species in particular, but if species tend to avoid divers, they might not be easy to identify when, well, visually, but then this environmental DNA offers this advantage compared to underwater visual senses.
Well, actually, visually, we always find them when we dive around the Zambia, but we can't find them very often in eDNA samples. We have it in the database, but we don't find them very easily in eDNA sampling.
Okay. Well, I could look up in which site we exactly found it and see if it's somewhere near where you are.
Yeah. Okay. Okay. I have another question. When you collect your samples in the MPA and around at 5 and 10 kilometers, did you collect them at the very same time, or was it 1 week or 2 weeks or 1 month later?
So, so our samples were always collected in duplicates. So we had this dinghy boat with two pumps filtering at the same time. And so our 4 replicates inside of each site were either conducted in parallel or half an hour apart from each other, because it took us half an hour to filter our sites. And then inside of each region, we try to always sample all samples the same day. So the reserves, the 5 kilometers and the 10 kilometers are typically collected in the same day, but then the different regions were well sampled on different days.
Okay, and do you did this only once at one season? Would you expect to have some differences if you would do this at different seasons, for example?
I think, yeah, we could definitely expect some differences from different seasons because, yeah, the presence of many marine species, I think many species go, for example, to deeper waters during winter and might not be at the surface. So we mainly sampled water from the surface and close and in undeep waters to really have these local detections. And I think maybe in winter we might detect way less species than we do in summer as these move to deeper waters. So I think sampling season definitely has an effect on yeah.
Okay, and I could remember you did some sampling more deeply when you came here. Did you analyze these samples? Just to know, because we couldn't talk about this previously.
Yeah, no, I did not. I did not analyze these samples.
Okay. And a more general question: what do you think about eDNA as a tool to help to choose marine protected area and to increase perhaps the connectivity between them at a regional and global scale? Do you think it could be a good tool for this?
So you mean to select areas that should be put under protection? I think we then need to know which species we want to target by our protection, but then definitely eDNA metabarcoding can allow us to look for the presence of certain species. And then if this is, for example, if there are some rare species we want to protect, we can look for their, well, their sites are most present in, and then target sites that are important to those species, and yeah, definitely understand where to place protection targeting specific species. I think that's my question, my answer. Yeah.
Okay, good. Thank you. I think the other questions have been already asked. So yeah, yeah. Okay, so I have no question anymore. So I would like to congratulate you again for your talk and your answer.
Thank you.
Thank you very much.
Thank you.
Thank you very much for that. It's my turn now. Can you hear me? Video conference, can you hear me? Yes? Robert, Oscar, can I? Yeah, okay.
Yes.
Because the microphone is off, so.
Ah, sinon, c'est lui, mais j'ose pas y toucher.
So thank you very much, Emilie, for inviting me to this defense, of course, but also.
Ouais, ça marche. Hein? Ça marche? Ouais. Pas bien. Ouais. Okay, so I really like your thesis work. It's really an impressive work, which is targeting both the species assemblage level and the infra-specific level. And you made a really nice job in integrating, in a way, the two approaches. So, so congratulations. It was really, really an interesting work. Many of the questions I have already been addressed, of course, but still I have a few. So, well, a very broad question to start first regarding the use of high throughput sequencing approach. From your results, from your work, it appeared that they are fantastic tools. However, I guess they are not devoid of limitations. And so, from your point of view and regarding your results, what for you are the most important limitations, or at least the points we should be very cautious with? And both for the eDNA metabarcoding work or the population genomics approach?
I guess one of the limitations is that while we try to target these entire assemblages, for example, with environmental DNA metabarcoding, we will still miss some species. And for example, between our different samples, even between different replicates, field replicates, or even different PCR replicates, we did find differences between those replicates where certain species were identified on one and other on the other. So I think one of the limitations is that it's, well, as any methods, it's not exhaustive. And we have to be careful to indeed understand that. So we have these species presences, but the abundance of the species, the absence of our species in our signal, for example, does not mean that the species is truly absent in the environment. So I think that could be one of the main limitations. I guess the same holds true for if we look at these SNPs at the species level and these genome-wide markers, is that we'll probably miss a lot of the fragments and a lot of the information that can be maintained. So I guess.
In both cases, you mentioned sequencing depth, sequencing coverage is likely to solve part of the problem, don't you think?
True. Yeah, true. But for example, for the environmental DNA metabarcoding, we really had a very, very high sequencing depth. And well, we did, if we look at, for example, I looked at saturation curves for our different samples in by this sequencing depth, and indeed we actually managed to reach saturation for our species detection by sequencing depth. So I guess that's...
Okay, and I was wondering about the variant culling too, because for sure, and you discuss a bit about that with Oscar, when we see this surprising result of finding siblings 2,000 kilometers apart.
Yeah.
And they are full siblings according to the results of your analysis. So it means that this is the present generation that migrate. How robust are those results according to you? Have you evidences, other kind of evidences, or I don't know, results of, I don't know, other data that, yeah, you see what I mean.
Results of this thesis that definitely merit to be further, investigated and maybe try to indeed corroborate with other results. I I don't think for example, other studies have found sibling relationships at this spatial distance before. So, and I don't know if with our current data, we might calculate other metrics, but I don't know if they will allow us they might allow us to then confirm this relationship between our individuals or not. So I think we definitely should push the analysis a little bit further, indeed, to increase our confidence in them.
I was wondering if, when you are modeling dispersal of larvae, you have always this very long tail. And if such distances might be predicted with the model, whether really at the right end of the tail. Do you know if the model, larval dispersal model, can predict such a distance?
I don't
even if very, very rare.
Yeah, I guess it could be very, very rare. Now, one thing, for example, our model didn't take into account is this larval movement and larval behavior. And I did find some studies showing that larvae of sea bream are capable of swimming quite fast and quite strong distances. So if these marine currents could take them up till, I don't know, like 50 kilometers, and then we can add this larval behavior to it. Maybe we are currently underestimating this capacity of larvae to disperse with our models by only modeling passive larval behavior. So I think, and I also think if they're siblings, for example, and they both dispersed in opposite direction, although this might be unlikely, that would increase the distance between them. Definitely, large spatial distance that we detected are closely related individuals.
So, well, when I read this result, I was just wondering maybe it might be a problem the calling, a kind of errors, due to the methods of calling, and well.
I think we definitely need to rule that out if we want to be how we would do that. But
we know that there are long distance dispersal events, very rare, and they can have strong impact, ecological or genetic impact, for example, for the colonization of new novel habitats. So that's why this result is really interesting. But for sure, at the moment, you have to be cautious with that because it's so incredible.
Yeah. I think I'm going to write that down. That verify.
Wow. Let me check because many questions, as I said, I'd been addressed. Oh yes, keeping on the results from the papers that you did not present during the defense about the graph theory, which was really, really interesting. I was just wondering. To which species it is applicable in the sense that it seems to be quite fixed over time. So how do you think it might be feasible to change a bit the algorithm and the way to model and predict this graph, this network, taking into account temporal events such as extinction or colonization, opening of novel habitats, or closure of habitats because of destruction of natural habitats. So where is the temporal aspects? Can you think to a way to include that? And are those temporal effects might have modify a bit the interpretation of your results?
Yeah. So I think we could definitely integrate that. For example, when we model this dispersal capacity between our different sites, we could then take out some sites that, for example, we know do not hold any of our populations or that are unsuitable habitats, or then we could add sites that are newly colonized. So I think by playing with these available sites in our model, we could definitely model how fluctuations over time and integrate it that way. And then see how this affects, the genetic results and how this cooperates with the genetic connectivity we found.
So, the point is that there are still a lot of discrepancies. It's really nice. You are refining a lot the prediction for the larval dispersal, and it's great to have this to implement this multi-generation, but still the discrepancies is there. And I was wondering if temporal effects, dynamics might be part of the explanation of the discrepancy. And also what else could still explain the discrepancies? You have a few sentences in the paper about that, but can you elaborate a bit about the factors that may still be there and explain the discrepancies between the two approaches?
So I guess in so many seascape genomics patterns or genetic patterns, we find a lot of what we call genetic patchiness and chaotic genetic patchiness. And I think those could be explained, for example, by high variability over time in recruitment fluctuations. So for example, if one population 1 year has high levels of
I fully agree.
productivity, for example, these connectivity patterns will vary. And if this varies over time, it could create these more chaotic patterns that we did not at all take into account. I think it would be difficult to know and track back in time how these fluctuations occurred in the natural system to then be able to represent them in our model. But I think they could account for a lot of the discrepancies we still found.
Yeah, any kind of post-settlement processes also have strong impact. So, as you say, the way recruitment is effective or not, and how it varies over time and over generation, of course, besides the behavior of the larvae, of course. And a last question regarding this part is, there is something maybe I did not understand properly, but there is something which seems to me a little bit weird. If I'm not wrong, I may be totally wrong, but if I'm not wrong, genetic data showed that sites in the Eastern and Western Mediterranean basin are more connected than sites that are within basin.
Oh, yeah.
Okay, and at the same time, you observe an overall isolation by distance pattern.
True.
And and so I was wondering, what is the explanation for this apparent paradox in a way?
That's a very good question. I actually didn't really think about that before. So.
Honestly, I have some hypotheses, but
yeah.
Perhaps it's maybe that basin are very large as compared to the distance, and that must, well, it could be a kind of artifact where the populations are and their distribution across the basin, or it can be driven by a subset. But this is something that would have, well, the paper is published, but it's something that it would have been interesting to point and analyze a little bit further, because it's a paradox at first glance.
Sure, just think paradoxes.
Oh, not that much. No, not that many. But well, if you find the answer just by curiosity, I would be interesting to know how you have this decreased connection within, and but still an isolation by distance pattern. It may be something interesting to consider for further studies.
Yeah, definitely.
Did you see the time? Because oh yeah, okay. I have a few minutes left. I don't have a lot more to tell, except, oh yes, yes. Of course, I, as the other members of the jury, I really like also the chapter about the MPAs and the species diversity and so on. So you already have extensive discussion with the other members about this part. So I think most of my questions were addressed. And I have a last question, a little bit more about the methodology. You made a fantastic effort, and really it's great to increase the number of reference, and we all know that this is a key in those for those analysis to be robust and reliable. So really it's fantastic. And you got 75%, if I remember well, of the species from the local pools. And again, it's more a question of curiosity. Did you make the analysis by reducing the number of reference available and looking at the results to know how is it really sensitive to the completeness of the database for the species you are targeting?
Yeah, definitely. We did not do that, but I think it's a very good idea to indeed assess, because we, for example, we compared the completeness of our reference database to then say that, oh, it's probably not biased because we have different similar proportions between all our species categories. But we indeed did not test if we take out some species, if our signal changes or if our results changes. We did not.
I think it might be worse, including to help other researchers not working with fishes like me, working with invertebrates most of the time, and for which we will never get that. And it would be really interesting with a group like fishes, for which you have the data, you have the experience, you have the markers, you have everything to assess how many we really need to get those general picture regarding species turnover, species assemblages. I'm not talking about targeting or identifying about the identification of a specific species. I'm really talking about the global patterns, alpha, beta, gamma diversity. How are they sensitive to the percentage of species that are covered in the reference database? And it might be really interesting to explore that. And my last question is, with that study, do you think that the temporal sampling could have provided with a different picture?
So you mean if we would have sampled the same reserve over time as well?
Yes.
I don't know. I think it would be very interesting to look at, and I guess that would depend on these higher trophic level species that we're hypothesizing drive this difference in species richness, whether their presence varies in MPAs over time or not.
Or the fishing pressure variation also, which is different over time between season. I was thinking about seasonal effects mainly. Are there seasonal effects? Are they because of the migration behavior, because of the reproductive behavior, because of the human behavior in terms of fishing pressure? And so, but you already answered that to another member of the jury. Temporal monitoring, pursuing this work over time would be really interesting.
And I guess I'd say we have a way now of assessing this temporal effect of fishing pressure, because we actually sampled the exact same reserves and sites again during the COVID-19 lockdown, where the overall fishing pressure on the entire Gulf of Lyon was very much reduced. And so that would, these results would maybe bring into light how very pressure, even if they're outside of the MPA, influence this effect we found inside versus outside.
Okay, yeah, sure. Thank you very much. Really, it was really nice to read this thesis and also to discuss with you. So now we are going to shift to your supervisor, and I think Stéphanie, you start first.
Thank you, Frédéric, and all the members of the jury for this nice discussion. So now I'm going to switch to French because I'm going to describe the story of the Emilie PhD. So because I think that for all my questions have already been discussed with Emilie. Alors d'abord, moi je voudrais rappeler le contexte de la thèse d'Emilie, donc qui est le croisement de l'écologie et de la génomique. Pour répondre à des questions, à des finalités de conservation, pour améliorer donc la conservation des organismes marins et également donc essayer d'améliorer les stocks de pêche. Mais donc ce croisement de disciplines, il repose donc sur la continuité d'une collaboration établie déjà depuis plus de 10 ans. Donc entre Marbec et le CEF, voilà. Donc Emilie, ta thèse est inscrite dans ce contexte. D'ailleurs là, je remercie Marbec qui a financé ta thèse. Du coup, mais toi, là, tu es depuis déjà plus de 4 ans dans ce contexte, puisque tu as commencé en master et tu as poursuivi avec ta thèse. Voilà, donc tu termines un peu une cohorte, puisque tu avais pris la suite effectivement d'Alicia Dalongi, comme l'a rappelé Oscar, donc, qui donc a continué aussi l'histoire. Et puis il y a eu Marco Andrello avant, voilà. Donc voilà. Et en fait, moi je dirais que le CEF, je pense que c'est quand même ta seconde maison. Alors Emilie, l'isolement non, surtout pas. Donc pourtant le Covid est passé là, mais dès qu'elle a pu, Emilie elle est revenue au CEF. Elle occupe l'espace au CEF. Et ça, j'ai envie de dire avec enthousiasme, bonne humeur et beaucoup de résilience. Hein. Dans une thèse, il y a des hauts et des bas, mais et voilà, Emilie, même dans les bas, elle remonte toujours. Voilà, donc merci pour cette bonne humeur. Ça va nous manquer. Voilà. Moi, des conseils, quelques conseils juste pour la suite, parce que c'est vraiment concentration, rigueur. Voilà, c'est ce que je te conseillerais, parce que tu sais finalement le faire, mais il faut qu'on te le rappelle. Voilà, donc c'est vraiment important pour la suite. Donc voilà. Et j'ai envie de dire un bravo pour ton oral. Voilà, vraiment, je trouve. Enfin, moi, j'ai vu donc une répétition il y a à peu près une semaine. Voilà, et vraiment, je trouve que tu as vraiment, bien pris en compte les remarques qui t'ont été faites. Voilà, tu as su rester concentré. Donc, du coup, tu sais le faire en fait quand tu. Voilà, donc. J'ai beaucoup apprécié. Voilà, donc merci pour ça. Et donc. Alors pour la suite, c'est vrai qu'on l'a pas évoqué. Donc voilà, tu as déjà commencé d'un poste d'ATER à Marseille. Hein, donc en fin de semaine, je crois, tu enseignes. Donc voilà, donc je suis bien contente parce que tu vas pouvoir. Finalement, je pense que tu sais pas encore tellement si tu vas partir en académique ou plus appliqué dans le domaine de la conservation. Donc cette expérience académique, elle va peut-être te montrer finalement, peut-être t'ouvrir d'autres voies. Voilà. Bon, en tout cas, je n'ai pas de soucis pour l'enseignement, là, finalement, à part que ça risque de prendre un peu de temps. Alors, attention à ton temps, hein, la gestion du temps. Voilà, donc, tu plonges, tu sais échantillonner, tu modélises. Donc, voilà, comment tu vas utiliser, encore d'autres choses, hein, je loupe, je raccourcis. Maintenant, à toi, voilà, de faire ton chemin pour la suite. Voilà, et donc, merci encore. Et je vais passer la parole à David.
Je vais continuer en français pour les commentaires généralistes et puis je passerai en anglais après pour des choses plus en lien avec les commentaires du jury et puis une question que j'aimerais poser quand même par rapport à la suite. Donc moi, je suis entièrement sur la ligne de Stéphanie quant à ton implication et ton intégration dans nos deux laboratoires. Tu as été moins à Marbec qu'au CEF, mais quand même à chaque fois... Voilà, la preuve, les gens qui sont venus te voir ici prouvent que voilà, tu as été toujours bien accueilli et tu as toujours bien interagi avec les gens de Marbec. Donc, merci pour tout ça. Tu es très, très bien élevé. Un petit clin d'œil à tes parents et à ta famille, très bien élevé. Et tu as toujours été très apprécié dans tes interactions. Et ça, c'est très, 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 très intéressant. Et après, je pense que tu as développé un profil très généraliste aussi. Hein. Tu étais capable d'aller du terrain sur un protocole d'échantillonnage jusqu'à l'interprétation écologique en passant par les pipelines bioinfo. Donc le problème de ça, ou en tout cas le backside, hein, c'est que tu n'es pointu sur rien, mais par contre tu es très généraliste, donc tu es capable de mener un projet de bout en bout. Et je sais que tu seras capable d'aller chercher des compétences pointues sur chaque aspect que tu as traité. Donc voilà, c'est un peu... voilà, en classe, les étudiants, il y en a qui sont hyper spécialisés sur un aspect. Toi, c'est le contraire, hein, on t'a mis directement sur quelque chose de généraliste, hein, de par ton financement et de par ce côté-là où on devait débroussailler un peu cet effet réserve sur la diversité via le prisme génétique. Donc chapeau, parce que c'est pas évident d'aller du terrain jusqu'à l'interprétation écologique, donc tu as su bien le faire, donc bravo à tout ça, bravo à toi, et c'était pas forcément évident de faire ça. Voilà, je vais passer en anglais après maintenant par rapport au jury. Frederick mentioned that the temporal seasonal effect should be strong, and now I think spring was not the best period because of the juvenile effect, perhaps. We know that juveniles can be everywhere. They don't care about MPAs or they don't care about human impact, and so I think the juvenile effect may blur totally the patterns we obtain. And to my opinion, if I have to redo again your sampling, I would suggest perhaps going more in September or October to avoid perhaps this juvenile effect. And nobody knows what can happen with juveniles. So what is your opinion? The juvenile effect may homogenize the pattern or may differentiate the pattern you obtain?
I guess it would depend if we consider that juveniles are, for example, also predated by the higher trophic level species. So if we have a lot of juveniles and that are under this higher predation pressure inside of the reserves, they might be found more outside. And then if they are not these cryptobenthic species, but these other species that as adults more benefit from this reserve protection, indeed it could kind of homogenize our signal because we would find the same species outside as juveniles and inside as adults. Or maybe we could find just the opposite and the juveniles find nurseries or protection inside. Well, I think it's the first option where juveniles might be more, might fare better outside where they're not targeted by this fishing pressure and might thus be found more outside. Whereas then as adults, they would be fished and find mostly inside, and it would homogenize our signal between the inside and the outside.
Yes, I agree. To me, we should perhaps see something more heterogeneous in September and October, and perhaps less species outside, because we found many commercial species outside in spring, but maybe we have juveniles. So it's perhaps why we found more species outside, because we have the commercial ones plus the cryptobenthic. So maybe it's September or October we would find something perhaps even more differentiated, but perhaps the same species richness. So I have a feeling now that the higher species richness outside is partly because we have many small and juvenile individuals outside in spring. That's something to test. My last question, you may apply to a permanent position in next spring if you want to become an academic one day, maybe yes. So since you have a very generalist profile, what will be your project within the next 5 years if you apply to a permanent position? Where do you want to go after you're at
where do I want to go? I guess I definitely would want to have a project where I have a lot of field work involved. I think this past year I've spent a little bit too much time on the computer and maybe not enough outside. And I'd love to more work on more conservation application research questions. So I think we're working more on how marine protected areas can affect our species assemblages, and maybe, as I said, go study these in more tropical seas and see if we find similar effects there or not. We know that cryptobenthic species are large drivers of these trophic, are very abundant in tropic seas, and it could be very interesting, for example, to see how they vary inside protected areas and outside, and what this means for protection. So I think I more further pursue these conservation applied research questions by combining again fieldwork and then the different methods and analysis afterwards, and be involved in all these steps along the way.
And how could you test your trophic hypothesis to explain there is lower species richness of cryptobenthic inside the reserves? How could you test that? Because we discussed that point many times together, and it's a shame that we couldn't test that very accurately. How could you test that?
I think at this point we could maybe use complementary sampling methods, really targeting, for example, these cryptobenthic species, and see if we target this same method inside of reserves and outside, and see if we find differences in cryptobenthics that would already illuminate one part of this big question mark we have left of what is the effect of these trophic interactions. Then I guess we'd have to better understand what the trophic relationship is between our cryptobenthics and then these larger species, and understand whether they're indeed predated by these other species, for example, in the Mediterranean, whether we have these trophic relationships between our larger species found inside and the cryptobenthics. So maybe, I know we discussed, for example, looking at stomach contact data of our, for example, sea bass and see if they indeed eat cryptobenthics or not, and then better understand these trophic relationships between the different species we have in our assemblages in general.
What about big cages inside an MPA as well? Do you think if we exclude, you see what I mean? We are in the middle of the no-take reserves. Do you think that if we put cages to eliminate the presence of the big predators, we may see the cryptobenthic coming back?
That could be, I hadn't thought about that. I guess that would be interesting to see. I don't know, it would be interesting because then it would maybe tell us that it's more of a behavior and an avoidance behavior of those cryptobenthics that really avoid the reserves because they know the predators are there. And the question is whether they will be able to understand whether this predation pressure is now kind of eliminated or not inside the reserves.
I have one last question about stomach content. You may imagine that it's not easy to identify cryptobenthic species in the stomach. How could you do to know the diet of big predators?
Well, we could again metabarcode the, I guess, and apply the same or similar metabarcoding protocols to then identify the species from stomach contact.
And once again, since we have a reference database for fish, it would be great to know the diversity of big fish diet. Could be, I think, it would be a nice project to sell within 6 months to your perhaps next position. So thank you for all that, and thank you, Frederic, and thank you to the jury for being here.
Thank you, David. So now it's time for the jury to deliberate. So I don't know, do you?
Maybe that we will ask to everyone to go out. How you people do that?
Okay.
Want to use this?
Désolé, on va vous demander de sortir le temps qu'on délibère.